portion of the lab for Lab K and water testing, looking at microbes in water. And this is assuming that you have already read through the lab procedure that you can find online. So now that you have read the lab, where might you find contaminated water? So in ponds and lakes of various types. Also, you may find contaminating water after flooding, which is seen in the bottom picture. So how does this fecal contamination end up in the water? Well, we've got lots of potential sources. So when we collect the sample, it's usually collected using a sterile bottle from the water, and in this case they're expecting quite heavy pollution, so the person is wearing gloves and a protective suit. Since the organisms are very dilute in the water, we need to collect at least 100 milliliters of the sample, and usually when you're out there you collect more than that, because 100 milliliters is really not very much. That way if something goes wrong, you've got more water as a backup. So since the organisms are so dilute, we're going to set up a vacuum filtration system. And on YouTube, this is showing a vacuum filtration setup with a chemistry setup in a lab. For microbiology, the equipment that you would use for this would be sterilized with an autoclave prior to being used, and the pore size is going to be specific to collecting the bacteria.
This is a demonstration on how to set up a vacuum assembly. A vacuum assembly is very important when we're filtering. We don't want to have to wait around for hours and hours for gravity to filter water through a filter. So what we'll do is we'll force the liquid through using a vacuum assembly. The vacuum assembly will need a filter, so we're going to use this filter. The problem with usually the filter and the filter paper don't fit each other. So what we'll do first is accurately cut this piece of filter paper to fit inside here. The best thing for you to do is to fold the filter paper in half, and then into quarters, and into an eighth. That way your cut is very simple. You cut right along the edge. Just from experience, I don't think that's quite enough. There we go. What we're looking for is a piece of filter paper that will cover all of those holes in our filter. Excellent. An important step here is to wet the filter paper with water, deionized water. So we'll wet the filter paper, put it down my filter paper. It's important to take a glass stirring rod and make sure that there are no bumps or crinkles in our filter paper. What the paper does is forms a seal over all of these holes. That way nothing slides past the paper. Next we'll fit our rubber stopper to our Erlenmeyer flask. The Erlenmeyer flask is a little different than the ones we've seen before. There's an outlet valve here. I placed it in this assembly to hold this down because this whole thing is kind of heavy and we'll get the Erlenmeyer flask tipping over. So using this ring stand to hold the flask down is going to be helpful to us. What we're going to want to do is take a piece of rubber tubing and attach it to our flask. And then we're going to attach the other to a sink. Not just any sink, but a sink that has a side nozzle on it. So we'll be not attaching here, we'll be attaching to this horizontal portion. And then to force fluid through, we simply turn the sink on. And you can see water dripping through here. Turn this off and we'll try with a colored solution. The solution I have here is simply food coloring and water. When I place it into the filter, we see nothing coming through. No dripping at all. Now just very slight dripping, not very much. But when I turn on my sink, the fluid flows freely and our solution is filtered in seconds instead of minutes.
So for our use here, we don't have that type of sink. What we set up is a vacuum filtration system using a vacuum pump. We also use a slightly different type of plug on the top here. It's still the same idea that you're creating a vacuum to pull the water through, but we don't have to cut the filters. We buy them that are specific to the size of the valve that we need here, because if you were to cut the filters, that would be introducing contamination into the process here. So the bacteria become trapped on the filter and the water passes through. So since we've taken the care to use sterile equipment, sterile forceps are going to be used to remove the filter with the organisms on them from the water sample. So you can see with this plug, the filter fits nicely right on the top. And then it's going to be placed on the appropriate medium for detecting fecal coliforms. And then we incubate it. And this is exactly the type of incubator we have in the back room. And we will incubate those at 37 degrees, which is body temperature, which is what fecal coliforms prefer to grow at. Being that they are in water, they can be grown at a slightly lower temperature, and they will still grow. It just takes a little bit more time. Usually we're trying to speed the process along a little bit, and 37 degrees is going to be the fastest temperature for growing them. So then we will observe growth. This one has the presence of fecal coliforms on it. You can see the eosin green sheen that is seen with the fecal coliforms. When we have organisms other than fecal coliforms on there, you'll notice that there's no green sheen present on here. So for your lab, your sample 1 is going to be contaminated water. And what you're going to want to do is count these little colonies here. And you can pause the screen on here to be able to count these and get the numbers that you need. You want to do your best to count them. I know that the picture is not exactly the most clear, but this is one that I could find that had a reasonable number to count to put in here. For your sample 2, this is going to be from a kiddie pool. This picture to the left is a really close representation to actually what we saw when I performed this lab with water from my niece and nephew's pool. And during the day, my niece had been out there in her pool, and we were out there, and I said, you know, I'll be back in a minute, I need to go use the restroom. And she goes, well, why are you bothering going in the house to use the bathroom? I just go in the pool. And obviously she just goes in the pool quite a bit from the amount of fecal coliforms that were present in their pool. Now, when you look at this, there is obviously way too many organisms to count, and so that's what you would indicate for the amount of coliforms on there. So the rest of your lab can be finished up using the articles that were included with your lab as well as the instructions, and hopefully this helps you make your next experience at a swimming pool with lots of kids more enjoyable.